Hello, everyone. My name is Dr. Elena Grintsevich. I am assistant professor in California State University, Long Beach. And today I would like to tell you about unusual mechanism of actin regulation. Specifically, the title of my talk is Regulation of Actin Dynamics by Mical Enzymes. First, I'm giving you an overview, the static image of eukaryotic cell, and this is a beautiful image of the fibroblast you can find in the review by Tatiana Svitkina. And as you can appreciate, and you can see those yellow arrows pointing to various types of actin-based structures. Those structures are very diverse, and they formed in the cell in the precise locations with the help of multiple actin regulators. And what I would like to point out here that this is a static image. It's showing you different actin-based structures, but in reality, in cell, actin cytoskeleton is very dynamic. And what I mean by this it means that actin structures assemble, disassemble, and remodel continuously. Well, first of all, for all of us to be on the same page, I would like to point out some key features of actin. And on your left, you can see a structure of actin monomer. And on your right, you can see a schematic of actin polymer. And as you can see, there are several regions in actin monomer, such as D-loop, DNase binding loop, hydrophobic cleft, and hydrophobic plug, which are involved in self-assembly interface. What this means that those regions are participating in actin-actin contact as monomer is transitioning into the polymer. And intuitively, we can think, and it would be true, that if there is some dramatic changes on those self-interaction interface, it can affect the properties of the resultant polymer, and that's what we will see in a few minutes as I'm talking about Mical family enzymes. Another key features which I want to bring to your attention is that actin polymer is polar. And what I mean by that two ends of the polymer, pointed end to minus end and barbed end, plus end, are functionally and structurally different. Second of all, what we need to understand very well for the purpose of today's conversation, that actin is a mixed nucleotide-state polymer. What do I mean by that? Every actin monomer has an ATP molecule incorporated into its structure. As this monomer transitions into a polymer, ATP hydrolyzes to ADP and inorganic phosphate. What is interesting, this inorganic phosphate is not getting released from the filament structure right away. It temporarily gets entrapped in the filament structure. This leads to the scenario when the filament has stretches of ADP-PI-rich subunits or proteiners, and we have stretches of ADP actin which already released inorganic phosphate. Why this is important? It's important because actin in different nucleotide-bound state has different properties and different interactions. And the last thing which I want to bring to your attention, since today we will be discussing actin disassembly mechanism, is what can trigger this transition from actin monomer to actin polymer. And you can see on this slide above this black arrow, which you can see in the center, that if I take actin monomers and I ionic strengths of my buffer and I add magnesium ions, I can transition my monomeric actin into polymers. Other way around, if I take a purified polymers and I remove magnesium ions, the lower ionic strength of the buffer, my polymers will fall apart into monomers. And this is something which we can easily manipulate in a test tube in vitro, and this is something which we're using for actin purification from different sources. But what we need to understand that in the cells, we don't have this dramatic transition of conditions, right? So all actin assembly or polymerization and disassembly takes place under the same conditions. And what's important to understand that those conditions favor actin assembly rather than actin disassembly. Therefore, there is a great interest in understanding how actin polymers are being disassembled under polymerization promoting conditions. Actin regulation system is extremely complex, and there are multiple groups of factors which regulate actin assembly and disassembly in cells. And of course, I'm not going to be talking about all these factors, but I just bring to your attention the focus of my talk today. I will tell you how actin disassembly can be orchestrated by Mical family enzymes. And I will tell you how action of those enzymes synergizes with one of the known actin disassembly factor called Cofilin or ADF cofilins. Also, I will discuss with you how this disassembly mechanism crosstalks to the effectors of actin assembly, filaments and profilin. What we need to know as monomers depolymerize and released from the polymer, they get sequestered by small actin sequestering proteins such as profilin or tyrosine beta-4. And this is very important to suppress spontaneous random assembly of actin polymers in random places in the cells. However, binding of actin to profilin is very physiologically important because there are multiple actin regulators which are evolved to bind the complexes of actin and profilin and utilize them to drive actin assembly. And I will discuss with you our most recent work which highlights how those mechanisms of actin assembly and disassembly crosstalk to each other. So first of all, what are Mical family enzymes and how we know that they act in disassembly factors? Those are very important molecules which are involved, as we know by now, to repulsive axonal guidance, synaptic structuring, dendritic pruning, development of heart and muscle, cytokinesis, as well as cancerogenesis and apoptosis. What is unique about those regulators, as opposed to most of the known actin regulators, that those are enzymes. Those are biological catalysts, more specifically flavin-containing monooxygenases. And what those enzymes do is very specific, and it's completely different from the oxidative damage we sometimes discuss in relation to oxidation of biopolymers. What those enzymes do, they oxidize actin on two residues, very site-specific and stereospecific actually. And those two residues are two methionines in DNA's binding loop of actin, and this is methionine 44 and 47. And for this reaction, you need to have NADPH as a coenzyme and molecular oxygen. What we know that this oxidation triggers actin disassembly in vivo and in vitro, and I will show you some data which highlights this. And also what we know now that this oxidation can be specifically reversed by methionine sulfoxide reductases. So how do we know that Micals are actin disassembly factors? What are the evidence of it? Well, this beautiful work here shown on this slide comes from Jonathan Thurman's lab. And they have this high-resolution system to monitor actin remodeling. Most specifically, this is Drosophila bristles. You can see this Drosophila fly in this figure, and in the blue rectangular, you can see the Drosophila bristles. So this system is perfect for studying actin remodeling because those bristles are very actin-rich. And so what we have here, that if we drive Mical overexpression in these bristles, we're getting this characteristic disordering of actin, as exemplified by those two panels on the top of the slide, where we can monitor actin assembly state using GFP-labeled actin. And so you can see that we can detect dramatic remodeling of actin in those bristles if we overexpress Mical enzyme. And what it leads to on the level of the phenotype, you can see that those bristles of Drosophila, they start developing those characteristic branches. And this is shown in the red frame over here at the bottom. And you can go ahead and compare it to the wild-type bristles, which normally do not have any branches, and they just long and a little bit curved. And so this is a very clear indication that this Mical enzyme regulates actin and disassembles actin in vivo. But what do we know about the mechanism, how exactly this happens? And the simplest explanation would be that upon action of this enzyme, we have production of some kind of polymerization impaired or functionally impaired actin, and it causes this kind of disordering. So what we did, we went ahead and we checked if this oxidized actin even capable of polymerization. And we were very surprised to find that it is capable of polymerization. What you're looking at here is bulk actin assembly assay, which is assisted by pyrene fluorescence. And what we have here, we have a fraction of actin monomers which covalently modify this pyrene maleimide. And they do fluoresce as monomers. However, as actin polymerizes and environment of pyrene probe changes, we see this increase in fluorescence multiple fold. And this gives us very sensitive assay to monitor bulk assembly of actin. And as you can see from this graph on the right, there are striking differences in the kinetics of assembly of unoxidized and Mical-oxidized actin at the same concentration. First of all, this Mical-oxidized actin is this purple curve. You can see this very long lag phase. And what this lag phase corresponds to, it corresponds to the nucleation stage, which is rate-limiting step in actin polymerization. And what it corresponds to 3 actin monomers coming together and forming a nuclei, which would seed polymer formation. And so as you can see, this process of formation of those nuclei are severely impaired in oxidized actin. So another thing which you can see very clearly that those polymerization curves are plateaued at different levels. And what this means that those actin preparations, unoxidized and Mical-oxidized, we call it Mox-actin, have a different critical concentration. This means that under polymerizing conditions, they will have different amount of polymer at 2.2 micromolar of total actin. And what does this mean in terms of what it indicates in terms of overall properties of these actins? Since critical concentration is a ratio of the rate constants of actin disassembly or depolymerization and polymerization, it means that the dynamics of actin is changed somehow by this oxidation. So here we wanted to go ahead and ask the question, what are the dynamic properties of this Mical-oxidized actin? And here I will show you two movies, and those are total internal reflection microscopy movies. And these movies show you real-time disassembly of actin polymers on the slide surface. So one will show you unoxidized actin, and another is going to be a movie of the oxidized actin. So let's just look at the unoxidized actin first. Time, some of those polymers are getting shorter, which is expected. However, in general, actin persists on the surface because its depolymerization rate is not really that high. However, if we compare this to depolymerization of Mical-oxidized actin, now I want you to take a look at the movie of oxidized actin depolymerization, and I hope you can appreciate how fast those filaments are disappearing from the surface. And what we see in those movies is very interesting and very unusual, because we see this catastrophic collapse of actin filaments, which was never seen for any form of actin before, and only was observed for the microtubules. So now what I can do, I actually can use these movies to quantify how fast those filaments depolymerize. And over here, you can see on the slide the quantification of such data. You can see those green and red traces, which would correspond to oxidized and unoxidized actin. Unoxidized is in green, oxidized is in red. And you can see that with oxidized actin, we can observe those extremely fast disassembly events, which we call catastrophic disassembly. Now I can go ahead and I can quantify how fast these processes are. And you can see that this catastrophic disassembly takes place with the rate of about 85 subunits per second, which is extremely fast. However, what we found, and what was very interesting to us, that we actually can artificially manipulate this disassembly rate if we mimic ADP-PI-rich state of the filaments, which we would call young or freshly formed filaments. And over here, if you look at this graph on the right, you can see that if we add inorganic phosphate to this Mical-oxidized actin, you can see that it depolymerizes much, much slower, and those catastrophic events disappearing. And also on the very right of this graph, you can see that if we add beryllium fluoride, which is a compound which mimics ADP-PI state of actin and binds very tightly to the filaments, you can see that oxidized actin filaments depolymerize rather slow. And that's what it tells us actually is that under cellular conditions, we might have a situation when filaments are oxidized, but they're fairly stable because they haven't released the phosphate yet. So now what we know that Mical is involved in the processes, cellular processes, when actin cytoskeleton needs to be disassembled or collapsed fairly fast. So how exactly does it work? Are there any factors which assist actin disassembly of a Mical-oxidized actin? So we asked, can actin regulatory proteins other than Mical alter dynamic properties of these filaments? And the first factor which we examined was ADF-Cofilin. And this is ubiquitous actin-binding protein, and it's essential for viability of eukaryotic cells. It induces specific morphological and mechanical changes in actin, and it has very distinct preference to ADP F-actin, to aged, older parts, stretches of actin filaments. And the reason why we were interested in this regulator and its potential crosstalk with Mical and oxidized actin is because this protein is known to affect the conformation of the loop, DNase binding loop, and that's the region of actin where those substrate methionine residues locate. So it was a potential that those regulators can crosstalk in vivo. And also, this is beautiful data from Jonathan Thurman's lab. You can see that you can have distinct colocalization of Mical and Cofilin in Drosophila bristles. So it is legitimate for us to ask the question if there is any synergy or potential crosstalk between these two regulators. And what we found was quite striking. When we formed unoxidized and oxidized actin filaments on the microscope surface and we added low concentration of human Cofilin 1, and you can see this on the very right panels, you can see that on the top panel it's unoxidized actin shown, and you don't see much of the disassembly, but what you can see that oxidized filaments pretty much disappearing from the surface within the mixing time, meaning that those two factors, oxidation by Mical enzymes and action of Cofilin, are synergistic. Another thing which was very surprising to us and very unusual in terms of the disassembly mechanism is that Cofilin gained the ability to sever actin filaments which are enriched in inorganic phosphate. And remember, I told you that this protein has a distinct preference to ADP-actin filaments which released inorganic phosphate, and it's normally not effective towards disassembly of the actin which is enriched in inorganic phosphate. But as you can see, if actin filaments are Mical-oxidized, then it allows Cofilin to sever those filaments very effectively, even in the presence of inorganic phosphate, as opposed to unoxidized filaments, as you can see on the top panels, which are not being severed and disassembled. And then we went back into the Drosophila model, and Jonathan Terman's lab performed this beautiful experiment when we downregulated and upregulated level of Cofilin on the background of Mical overexpression in bristles. And so what you can see on your left, that this characteristic phenotype, branching phenotype, is disappearing if we downregulate Cofilin. However, you can see very severe alterations in the bristle morphology if we upregulate Cofilin and Mical at the same time, which is indicative of those two proteins synergizing in their action, not only in vitro, but also in vivo. And based on this, we proposed our first working model of this new redox-dependent mechanism of actin disassembly. We showed that this mechanism allows Cofilin and Mical to disassemble filaments independently of their nucleotide-bound state. And the way how it works is that Mical is getting activated. It's normally auto-inhibited to allow for very localized effects. And for instance, here it's shown that it's activated downstream semaphore and plexin pathway. It oxidizes actin filaments, and what it leads to, and I'm not showing this data here, that those oxidized filaments have higher affinity to Cofilin. So Cofilin binds better to those filaments, which leads to enhanced disassembly in different nucleotide-bound state, and leads to actin disassembly and depolymerization. And so when we proposed this model, what was interesting to us and what we wanted to determine, what we wanted to know, what is the fate of this oxidized actin monomers. So we know that oxidation can be reversed enzymatically. However, what we wanted to know if actin assembly factors or assembly assistance factors can drive this oxidized actin back into polymeric structures. And what would be those factors, and what are going to be players here? As we know, as soon as actin depolymerizes, it will be bound to profilin, and we showed in vitro titration assays that Mical-oxidized actin monomer can bind to profilin, and then those profilin-actin complexes can be potentially utilized by actin nucleation and elongation factors. So we wanted to know, would it be the case for Mical-oxidized actin? Can cells repolymerize Mical-oxidized actin in the complex with profilin? And why this is of any interest is because of our data showing that even incorporation of small percentage of this oxidized monomers into actin polymers can really alter their properties. And if this oxidized actin after depolymerization can be driven back into polymers, that can result in long-range effects, which would affect cellular dynamic and function. So what we did, we took Mical-oxidized actin-profilin complexes and we asked, can we trigger their polymerization? And what this graph is showing you, and again, this is pyrene-assisted bulk polymerization assay, you can see that we cannot really trigger polymerization of these complexes. Even if we add into the solution preformed F-actin, which forms as seeds, and that's what seeds means in the legend of this figure. As opposed to unoxidized actin, which polymerizes very well in the presence of seeds, because now inhibition of nucleation by profilin is bypassed, and it polymerizes, however, slower without seeds as well, and this is going to be a green trace. We said, okay, maybe actin assembly factors such as filaments can perform this function. They can take those oxidized actin complexes with profilin and build the polymers out of them. So how do we check for that? And so this is the schematic of this formant protein. There are 15 mammalian formants. This is just a basic schematic of part of the formant structure responsible for a regulation of actin dynamics. You can see that it's a donut-shaped homodimer, and this homodimer can nucleate actin filaments, and it stays processively bound to the barbed end of the filaments. And this structure has a capacity to bind actin-profilin complexes because those extensions, which are shown in blue, contain polyproline tracks, which interact with profilin or profilin actin, enriching actin concentration near barbed end and accelerating polymerization. So what we wanted to know, can such factors reassemble Mical-oxidized actin, which was complexed with profilin? And what this figure indicates, and this is our most recent data, that no, we cannot really trigger polymerization of this oxidized actin-profilin complexes, even in the presence of formants which act on nucleating and elongating factors. As you can see on the right, all the traces containing Mical-oxidized actin or Mox-actin appear to be flat. It means there is no polymer formation in this system, as opposed to unoxidized actin, which polymerizes very rapidly in the presence of profilin and diaphanous formant 2, MDIA2, and also in the presence of F-actin seeds added. And just to tell you that we also went ahead and we checked 2 other formants from different classes to make sure that this is really a phenomenon which true for different formant family proteins, and we observed the same thing. We observed the situation when Mical-oxidized actin-profilin complexes cannot be utilized by these formants. And just to confirm that that's indeed the case, we again did curve microscopy, and we attempted to grow actin unoxidized, which you can see at the top panel, and Mical-oxidized bottom panel from labeled unoxidized seeds in the presence of formin and profilin. And you can see those beautiful long filaments formed in unoxidized sample, and you can see that there is no filament growth in the Mical-oxidized sample, since Mical-oxidized actin was complexed with profilin. And another very important observation we made is that this binding to profilin is not just arrests the assembly of this Mical-oxidized actin, but it also destabilizes already existing filaments. And here you can see the assay where we add an increase in concentration of profilin to preformed polymers of unoxidized and Mical-oxidized actin. And then we determine, usually using high-speed centrifugation, the amount of filamentous actin in the pellet in a supernatant. And so what you can see that Mical-oxidized or Mox-actin is destabilized by profilin to much higher degree than unoxidized actin. And so this data allowed us to go ahead and revise our model, our working model, and now we can say that this Mical-oxidized actin, because of its binding to profilin, is restricted in its ability to reassemble into the polymers and form either Mical-oxidized actin polymers or mixed copolymers, and it's due to its binding to profilin and inability of formants, and we'll also check INAVOSP as a actin elongation promoting factors, inability of these regulators to utilize those complexes. And at the end, I would like to acknowledge help from my collaborators. All the cellular work, Drosophila work was done by Jonathan Kerman's group. And I also would like to acknowledge Emil Reisler's group and Hong Zhao group. And Hong's group worked with us on the structure of Mical-oxidized actin filaments. And I would like to acknowledge NIH funding to my collaborators, and thank you all for your attention.